Hey everybody, we're going to look at bulk fluid transport at the capillary, and in your course pack this information begins on page 58. So we've talked about capillaries being very thin-walled vessels. In fact, they only contain tunica intima, and therefore it is easiest here in the capillary for molecules to be transported across. Sometimes that transport may include diffusion, such as smaller substances or lipids. We can see also active transport occurring across the membranes of those cells that line the capillary. But sometimes we actually see filtration occur, not across the walls of the cells lining the capillary, but rather in between those cells in holes called fenestrations or pores. And that's what we're talking about today, is how fluids move through those pores of fenestrated capillaries. Just a reminder that capillaries are single cell layer thick. They are composed only of tunica intima, and you can see in this picture that diffusion may occur across the cell membranes, while we are specifically talking here about the bulk flow of water and dissolved substances between those cells in pores called fenestrations. So this just kind of gives us a viewpoint of how those molecules can move from capillaries to surrounding fluids called interstitial fluid and into surrounding cells and tissues. So our focus today is mainly going to be on how fluids move from capillaries into the interstitial space, into cells, and then back into the capillary. So you may wonder why capillaries are the topic of our discussion. Why not arteries, since they have the highest pressure? Well, capillaries are certainly thin-walled, unlike any other kind of vessel that we've discussed. And capillaries are also under some hydrostatic or blood pressure. So you can see in this image that capillaries don't have zero pressure like the vena cava. Instead, they have a small amount of pressure, and that's all it takes to move fluids out of the capillary, is that small amount of pressure. Just a review of some fluid compartments. In the past, you may have learned about intracellular fluid, or ICF. This accounts for around 2/3 or 66% of the body's fluids. That's about 20 plus liters of fluid found inside of our cells. Extracellular fluid accounts for about 1/3 or 33% of the body's fluids. And of course, those fluids are housed in the blood plasma and interstitial fluid primarily. So you may recall that plasma is the fluid found where? That's fluid found inside of a blood vessel. That's of course in the blood. And plasma is the liquid portion of the blood that contains solutes, hormones, waste products, nutrients, and so forth. Interstitial fluid, or IF for short, is the fluid found in between cells and tissues. It is outside of the bloodstream, outside of the capillaries that we're going to discuss today. And of course, interstitial fluid does account for a good portion of the fluid in our body's fluids. Not as much as intracellular, though. So in order to understand this conversation today, we're going to be talking about two battling pressures, osmotic pressure and hydrostatic pressure, and we'll talk about this particular image and how these pressures manage the fluid movement out of the capillary and the return back in. So let's talk about hydrostatic pressure. First of all, the term hydrostatic means water pressure, and this is the same as blood pressure, or BP. This particular pressure is created by the presence of fluids wherever it's located, and it pushes fluids from areas of high hydrostatic pressure to areas of low hydrostatic pressure. Okay, so I've underlined and bolded some terms here that are really important to remember. Hydrostatic pressure is based on fluids. It is a type of pressure that pushes fluids from high to low pressure. By contrast, osmotic pressure should sound familiar. You remember osmosis is not, of course, blood pressure, but instead it is created by the presence of solutes and proteins in fluid. So sometimes I'll say salts in exchange for solutes, but it is those solutes and proteins that create a pull of fluid toward the highest solute concentration. Keep in mind that not all solutes or proteins can move across membranes to their area of lower concentration. Sometimes they are stuck on one side of a membrane, and instead of them moving, they attract fluid to their higher concentration. So you'll notice here, this particular pressure, osmotic pressure, pulls fluids toward the highest solute concentration. Let's further talk about two types of hydrostatic pressure. Hydrostatic pressure can be represented by HP. The one we're primarily focusing on today is capillary or blood hydrostatic pressure, known as BHP for short. This is the pressure of the fluid inside the capillary that pushes against the walls of the capillary. Keep in mind that hydrostatic pressure is based on fluids, and it is a pushing pressure. In fact, BHP is pushing fluids out of the capillary into the interstitial space. By contrast, there's a small pressure in the interstitial fluid called interstitial fluid hydrostatic pressure. Some textbooks call it HP with a subscript of IF. Others call it IFHP. So however you abbreviate it, it's still a small amount of pressure in the interstitial space. Keep in mind that hydrostatic pressure is a pushing pressure, and this pressure is pushing fluids back into the capillary from the interstitial space. But it's a small pressure, and it's not going to be able to battle against that great hydrostatic pressure coming from the capillary. Now, osmotic pressure is represented as OP, and the one we're going to focus on here is called capillary or blood osmotic pressure. Some books call it BOP, some books call it OP with a subscript of C. We'll just stick with BOP for simplicity here. Now we know that this pressure, osmotic pressure, is created by solutes and proteins present in the blood, and all osmotic pressure creates a pull of fluids toward the highest salt or solute concentration. Now there is a small amount of interstitial fluid osmotic pressure. There are some solutes and proteins in the interstitial space, but it doesn't have a great influence over this overall push and pull that we're getting ready to look at. So just to summarize, here are your four pressures. The two that we're going to focus on are BHP and BOP, so I'm going to put a star beside those. Those are the two capillary pressures that either push fluids or pull fluids. And of course, there's some numbers there for us to look at, and we'll talk about those on an image in just a moment. So you're not required to memorize these pressure values down here at the very bottom. Instead, we're going to talk about filtration pressure in just a moment, and that's the number you'll want to look at. I would not advocate memorizing this chart. Instead, I would encourage you to be able to talk about it using the next image. So you may want to grab a piece of paper or the image on the bottom of page 58 and just label with me as we work through this picture. We're going to talk now about how fluids are moved out of a capillary and into the interstitial space based on pressures. So first of all, at the arterial end of our capillary, we have two pressures. We have BHP and we have BOP. So blood hydrostatic pressure on the arterial end is fairly high, around 35 millimeters mercury. Okay. And at the arterial end, we're looking at blood osmotic pressure of around 26 millimeters mercury. All right, so one of these pressures pushes fluids, the other is pulling fluids. We know, of course, that blood hydrostatic is pushing fluids and blood osmotic pressure is pulling fluids. So we'll just keep that note there to remind us. Now, the higher pressure wins the battle. So 35 is our highest pressure. It will then overcome the osmotic pressure and force fluids out into the interstitial space. So there are fenestrations or pores between the cells of the capillary. That's where the fluid is forced from. It's forced through those fenestrations and first here into the interstitial space. And then, of course, it can continue on into the cells. So imagine a tire with a hole in it. It eventually leaks and loses so much pressure that it becomes flat. And of course, blood hydrostatic pressure works the same way. We're losing so much fluid to the surrounding interstitial space that we're going to see the fluid, or the pressure of that fluid, eventually reach a very low number, around 15 to 17 millimeters mercury. The osmotic pressure, though, is stable because the amount of solutes and proteins inside the capillary tends to remain stable at around 26 millimeters mercury of pressure. Like I mentioned a few minutes ago, the highest pressure wins the battle. So here, osmotic pressure is the winner, and it pulls fluids back into the capillary from the interstitial space first, and then, of course, fluids are pulled from the cells to the interstitial space, that fluid there called interstitial fluid, and then back into the capillary. So it's a way for us to force fluids and nutrients and solutes out into the interstitial space Most of it is reclaimed back into the capillary. In fact, around 90% of what we filter out is going to be reabsorbed. So let's name these processes. At the arterial end of your capillary, the process of forcing fluids out is called filtration. That's because we're pushing fluids from high hydrostatic pressure to low hydrostatic pressure. And it is hydrostatic pressure that's responsible for filtration. Now, due to the drop in hydrostatic pressure, at the venual end, that osmotic pressure is now able to overcome the hydrostatic pressure, and at the venual end, we're now able to see reabsorption occur. So, like I mentioned, about 90% of the fluid that is pushed out is reclaimed back into the capillary. The remaining 10% cannot stay in the interstitial space. If it were to stay there, it would cause a swelling condition that we call edema. Edema technically means extra fluid in the interstitial space. Now, if fluid hangs out there long enough, that fluid will eventually make its way into cells, and cells get bigger and they bloat. Okay, so I want to point out here that edema specifically means swelling in the interstitial space. It does not necessarily mean that cells are bloated, although that would be the next step. So how do we get rid of all this extra fluid? We pick it up in a lymphatic vessel that travels in between these cells and tissues, and that's how the other 10% of fluid gets picked up. That fluid is moved into the lymphatic vessel. That's a topic we'll hit in the next chapter. So this particular image shows net filtration pressure. You're not required to be able to do a net filtration pressure calculation. The reason I'm showing this to you is so that you can see that all four pressures are taken into account when that calculation is done. The two pressures in the capillary, hydrostatic and osmotic, and the two interstitial fluid pressures, again, hydrostatic and osmotic, account for this net filtration pressure. The one thing I do want to draw your attention to is this. Osmotic pressure in the interstitial fluid is around 1 millimeter mercury, and hydrostatic pressure in the interstitial fluid is around 0. So those two pressures contribute very little to the overall calculation for net filtration pressure. So when we do the overall calculation, we get a net filtration pressure, that is the overall pushing value at the arterial end of a capillary at approximately 10 millimeters mercury. It is a positive pressure. That tells me that fluids will be pushed out of the capillary and filtration will occur. When net filtration pressure is negative, such as what you see at the venual end of a capillary, we expect for reabsorption to occur. And in fact, net filtration pressure at the venual end is -8. So these are two values that you'll want to make sure that you understand and know. So to summarize what we've talked about so far, when fluids are forced out at the arterial end of the capillary, it is done so by the higher blood hydrostatic pressure. And when the fluids are pulled back into the capillary, it is again for that blood osmotic pressure. 90% of the fluids that we are pushing out do return to the capillaries at the venous end. The other 10% are picked up and returned to circulation by the lymphatic system, and that will be our next chapter. Just want to mention this particular condition is called a kwashiorkor, and it results in a large belly that's filled with fluid due to a lack of protein in the diet. Because we're talking about a lack of protein in the diet, do you think these children experience a loss of hydrostatic pressure or a loss of osmotic pressure in the capillaries? Well, protein is related to osmotic pressure, so these children experience a lack of osmotic pressure in the capillary. Increasing the protein in the diet can help with this situation. This is also ascites, and we see this in people who have, say, chronic heart failure. We see it in people who are alcoholics. There's a number of reasons for this condition, including liver disorders. And here we see either increased hydrostatic pressure or what else? Decreased blood osmotic pressure. So too much force of fluid pushing out into the interstitial space, or not enough protein to be able to reclaim it back into the venual end of those capillaries. So this accumulation of fluid around the belly is called ascites, and it's associated with a number of conditions. Now, there's a worksheet on page 59 for you to try, and once you have your opportunity to work through those questions, check out the answers that I will post for you in D2L.